Thank you. So, as the first speaker of the day, I thought I would mainly focus on what I see is the problem with our current understanding of neurocognitive aging. That is the fact that it's based on these weird samples, that is, studies conducted in Western, educated, industrialized, rich, and democratic nations, run by weird labs, which leads to some weird results and a lack of understanding of how different groups of people experience aging. So just a quick word about me. First of all, I'm an associate professor and Canada Research Chair in the Cognitive Neuroscience of Aging at Brock University. I completed my PhD in psychology at the University of Toronto with Lynn Hasher and Cheryl Grady, followed by postdocs at the Cambridge Centre for Aging and Neuroscience in the UK and Harvard. I would say that I am a cognitive neuroscientist. My research aims to address how does normal human aging affect attention and memory and the neural processes that underlie these functions. I use behavioral measures, including eye tracking, but also EEG and fMRI. And I tend to use a cross-sectional approach, testing extreme groups of younger and older adults at a single point in time and comparing them. Some main findings from my work are showing that age-related declines in attentional control lead to greater encoding of distraction and the formation of excess associations, so finding that we've referred to as hyperbinding, and also altered encoding of more naturalistic stimuli, such as movies and narratives. Now, one issue with most of my research to date and many others in the field of cognitive neuroscience is that it's based on convenient samples of older adults who volunteer to come into the lab. They opt in to our research. The problem with that approach is that these samples are really weird. So this is a seminal paper published in 2010 by Joseph Henrich and colleagues, where he coined the term. It's now been cited over 14,000 times. And in it, they point out that much of psychology is based on convenient samples of undergraduate university students from these Western, educated, industrialized, rich, and democratic countries, mostly in North America. And we then, as a field, extrapolate these results to make conclusions about human nature in general. And the problem with that is that, these findings don't hold up. These are not representative samples. They show that, a lot of our findings don't hold up across different cultures. And even more importantly, the fact that researchers from these weird countries are influenced by our own experiences and culture in the research questions that we choose to address. And aging studies are really no exception. In fact, I would argue that there may be even weirder, right? So for instance, in a review paper by Dotson and Duarte, they surveyed two leading cognitive neuroscience journals between the years of 2018 and 2019, which yielded 208 articles focused on aging. Within those papers, race and ethnicity data was reported in only 14% of the papers, and information on socioeconomic status, such as education or income, was reported in 18% of the papers. Less than 1% of these papers examine these factors as predictors of their neurocognitive outcomes. And among the studies that did report the demographics, the race and ethnicity of their participants, the majority of the participants were white. And in my experience, older adults who come into our lab to take part in research are also highly educated and relatively wealthy. They get themselves to the lab, they drive in their own cars. Like, that's a sign of wealth, right? And why is this the case? Well, the primary reason is our recruitment methods. We rely on these convenient samples. We put an ad in the paper. So it's like whoever's reading the paper and is like, oh, I think I'm going to go take part in this study. Or we recruit from alumni from our universities. There's also historic mistrust in a lot of marginalized communities because of past exploitation of these groups by science. People from disadvantaged backgrounds might also have poor access, like I said, be unable to drive to your lab, and also a lack of time. If you're an older adult who's still working to support yourself or your family, then you don't really have spare time to take part in this research. And finally, it's this lack of consultation. We've already heard this mentioned, poor consultation with these communities to ask them, what sorts of questions do you see as a priority for our research? And a lot of this is due to the fact that these labs are weird labs, right? We're all in these Western countries. There's a lack of diversity among principal investigators. And as I say this, I'm acutely aware of the fact that I'm a white woman named Karen, no less, speaking to you about this from my like middle class upbringing. But I've also been trained in an academic system that has lacked diversity for years. The questions I address are influenced by my supervisor's questions and influenced by the supervisors before them. So, we're sort of like maintaining this, this stream of questioning that might not be exactly what other communities think is important. Like I said, it affects the questions we ask and the approaches that we take. It also leads to some pretty weird definitions. So for instance, for today's meeting, we aim to address the question of whether there's specific sex, gender, and diversity related experiences that may promote or hinder resilience and healthier cognitive brain outcomes with aging. And implicit in this question is this emphasis on maintenance of youth-like cognitive functioning. That's the gold standard. Oh, you want your memory to be like a younger person's memory, which is ignoring a lot of the advantages that might actually come with aging. But this is also part of a broader, weird definition of what successful aging actually is. So in this seminal paper by Rowan Kahn, they outlined their model of what they think successful aging is. I'll try to put my pointer on here. Where they see like the central or the most important factor is, first of all, avoiding disease and disability, followed by high cognitive and physical function and engagement with life. And this medicalized definition of successful aging has been criticized, first of all, for its focus on function, right? The need to maintain good cognitive and physical health. But also it puts a lot of onus on the individual. It's your responsibility whether you're aging successfully, not due to any sort of structural determinants in your ability to do so. And also it's been criticized for its poor applicability outside of these weird samples. So for instance, Pace and Grenier reviewed the literature on how older adults within North American Indigenous communities define successful aging. And they came up with 4 main components to this definition. The first was health and wellness, right? Similar to the other definition. But in this case, this was seen as desirable, but not required for successful aging. Second component was empowerment and resilience. So this is the more emotional or attitudinal component to success, maintaining your connection with the land, spirituality, and things of that nature. Engagement and behavior. So teaching future generations about your culture was seen as an important role for elders. And also one's connectedness, maintaining relationships, family, community, social support. These are all also equally important aspects of aging successfully. So even somebody who's experiencing physical decline could be said to be aging successfully if they're able to maintain these relationships. And importantly, this is not a hierarchical organization. It's not that health and wellness is the pinnacle here. These are all interconnected and they are seen as influencing each other. So I'm not saying that determining the lifestyle and demographic variables that contribute to good cognitive health shouldn't be our goal here today, but we maybe need to keep in mind that just one piece of a larger puzzle of successful aging. And that resilience for some communities may be defined differently. And we maybe need to reconsider our aims based on consultation with communities, the communities that we wish to study, an issue that I'm going to return to later in discussing potential solutions. So all of these weird samples end up leading to some weird results that don't necessarily generalize to other groups of people. And it could be that this is contributing to the replication crisis that we see within psychology. So I'm guilty here as well, it also applies to my work. So it's not important what this study is about, but this is our attempt in a collaboration with Carolyn Clark and Christy Multhop. We aim to replicate a study I had from 2014, now in an online sample with English-speaking adults and Spanish-speaking adults with all the materials translated into Spanish. And these studies are now run online. And we can see a replication or a relative kind of a replication of the original pattern in the English-speaking adults, but we're not seeing the same effects with the Spanish-speaking adults. Now, we're not quite sure why that's the case, but it's just an example of how when you try to take findings from one group of people and extrapolate to another, they might not apply. We also know that there's a higher incidence of dementia in some racial and ethnic minority groups, but if we don't study those groups, we're not going to know why that is the case. And we also have, everyone's probably familiar with this figure from the report of the Lancet Commission about modifiable risk factors for dementia. But we don't know, do these apply to marginalized communities or even if they're feasible? It's all well and good telling somebody to eat like a Mediterranean diet, but if they can't afford or access fruit and vegetables, it's not really going to be a good solution in that case. Okay, so I've talked about a lot of problems. What are some possible solutions? Well, as a bare minimum, we need regular reporting of race and ethnicity data and socioeconomic status of the samples that we test. We can't determine if things are changing if we don't report this information in our papers. We need greater recruitment of non-weird samples. And if we want to look at these factors as predictors, and I'm sure we'll get to this in the method section, we need big N's. You can't really look at it if you've got small samples. So you might think, oh, okay, testing online might be a possible solution. It's really easy to collect data quickly online. Well, unless you do a targeted sampling like we did in that collaboration I just talked about, here's another set of data from this series of large studies that we ran during the pandemic through a Qualtrics panel. And I just point out that in this sample, since we didn't set any requirements for diversity, the majority of the older participants especially are still the same white older adults. They're all very highly educated, have high occupational attainment. So it's it's kind of the same samples that we're getting in the lab. Alternative recruitment methods might be warranted, such as the opt-out method used by the Cambridge Center for Aging and Neuroscience. The way people were recruited for this study was in the first stage, when they tested 3,000 people, they did an in-home interview. And these people were recruited through doctor's surgeries. They just got names and addresses of people through the GPs and sent out letters saying, we're going to come to your home and do this interview. If you don't want to take part, tell us no. Of course, when they get there, there's an informed consent process and everything, right? People aren't forced to participate, but it ends up getting you a much wider swath of the community than if you just post an ad and say, who wants to take part in our study? At that home interview phase, they measured cognitive and demographic variable simple cognitive measures, and then 700 people from that sample were recruited into the lab, equal numbers of men and women, to perform 7 hours of cognitive tasks, plus an hour of MRI and 30 minutes of MEG scanning. So if we take a little look at some rough demographic variables relative to census data from the Cambridge area, we can see, at least at the home interview phase, we're seeing a better correspondence than you typically see in psychology studies of, people who have no sort of qualifications. Well, this is Cambridge, so they're mostly white, and that's true of the sample as well, but also people who are unemployed. We see similar numbers, but these drop off when you then look at who qualifies to come into the lab for MRI, and I don't have time to go into why that is the case, but you might be able to guess. We also need greater collaboration with researchers from other institutions and countries, such as with the Global Brain Consortium, which is using EEG in populations all around the world. It's a much more portable kind of technology and affordable technology for other countries. We also need to continue our recruitment of diverse trainees who will hopefully go on to become diverse principal investigators and maybe change up the questions that we're asking. These students and RAs might be able to help us with recruitment and hopefully minimize stereotype threat in the lab. I don't have time to go into this whole off on a tangent about this, but older adults are already in a position of confirming age biases about memory performance in the lab. And this might be particularly the case if you're from another group who have negative stereotypes about your group and you're interacting with a young white RA in the lab. We also need greater consultation with the community. So you might be familiar with the phrase, nothing about us without us. And this is the notion, right, that you need to be informed by the priorities of the groups you wish to study. You know, asking what sorts of questions we should be addressing. And I'm kind of running out of time, so I won't go into that too much. But also, you know, are these current lifestyle recommendations that we have even feasible? And finally, this, you know, like we're trying to do here in this meeting, we need knowledge mobilization. The findings need to be translated back to the communities from which they came. You know, just come in, do research, and leave. Okay, that's it from me. Thank you.